Next, specification number 2 states, draw diagrams to represent observations of the animal and plant cells examined above. This guides students to create diagrams reflecting their observations of these cells. We will focus on creating drawings of the animal and plant cells previously observed, which will serve as references for students as they make their own diagrams. This activity offers visual aids for biology or science classes and helps students refine their scientific illustration skills. Comparing the microscopic drawings of plant cells and animal cells reveals several key differences. The plant cells illustration depicts some adjacent cells with double outlines, indicating the presence of a cell wall, a defining feature of plant cells. In contrast, the animal cells section shows some clustered cells outlined with a single line, reflecting the absence of a cell wall, typical of animal cells. This visual comparison highlights the distinctions between the two cell types and serves as a helpful reference for students to create more accurate diagrams from their microscopic observations. When examining plant cells under a microscope, students should draw a few adjacent cells from the observed tissue, using double outlines to indicate the cell wall, a key feature of plant cells. Emphasize the importance of cell walls in their structure and function by ensuring the plant cells appear organized and tightly packed. The drawings should be neat, with the cells depicted in a more regular, rectangular shape to convey a structured and rigid appearance. The diagram should be free of shading, coloring, or any additional details. Use a sharp pencil to avoid overlapping lines or confusion. Label everything neatly without crossing any organelles, ensuring that all visible organelles are accurately represented and labeled. Specifically, in the onion epidermal tissue, clearly depict the cell wall, nucleus, and cytoplasm. For the observation of plant chloroplasts, draw and label a large central space for the cell sap vacuole, with many small, round chloroplasts positioned at the corners. Use a double outline to represent the cell wall, making sure it appears slightly curved while maintaining a rigid and organized shape. Additionally, note that the cell walls are interconnected through spaces in between called plasmodesmata, which link neighboring cells. In contrast, animal cells exhibit distinct characteristics when viewed under a microscope. After observing animal cells, students should draw the adjacent cells clustered together, outlined with a single line to emphasize the absence of a cell wall, which is a defining feature of these cells. This outline effectively illustrates this absence, serving as a clear guide for students to accurately depict animal cells and highlight how they differ from plant cells. Additionally, note that these cells are not tightly packed, resulting in irregular shapes and overlapping arrangements. Use a sharp pencil for a tidy drawing, capturing visible features such as the nuclei, cell membrane, and cytoplasm in cheek, skin, and liver cells. You can observe how they cluster together to create a tissue. Clearly label each part and refrain from using any shading or coloring.